Hi guys, today we will be discussing about the protein structures. Basically, we will be discussing about the primary structure, secondary structure, tertiary structures, quaternary structures of the proteins. So let's start with that. So proteins that exist in various forms that may exist in primary structure form and secondary structures form. These are the more dominant forms and comparatively simpler than the protein that may exist also in the tertiary structures forms and the quaternary structures form. We will be discussing in details all of this in the next few slides. So when we talk about the primary structure of the proteins, that consist of a linear sequence of amino acids, and these amino acids that are linked together by the peptide bonds. In the previous lecture, we have seen the structures of different amino acids: alanine, arginine, asparagine, aspartic acid, cysteine, and we have also seen the classification of these amino acids. So all of these amino acids that are linked together, uh, these are the amino acids you can see. These are actually linked together to form the primary structures of the proteins. So look at this one, how that are linked together. Look at this one. This is actually the peptide linkage, C double bond O NH. Basically, these are stabilized by the hydrogen bonding, this structure that are stabilized by the hydrogen bondings. So look at this one. This is the first amino acid. This is the second amino acid. This is the third amino acid. So these amino acid that are linked together by the peptide linkage. This is the peptide linkage. This is the peptide linkage. So what is this? CONH. So the first amino acid, the carboxylic group of that amino acid that is linked to the second amino acid, and for this second amino acid NH group that will be involved. So CONH, that will be a peptide linkage. Same is the case with here in the on the second amino acid, this carboxylic acid is involved, and for the third amino acid, NH amino group is involved. So what is the whole configuration of that? On one end there is a nitrogen end, and on other end there is a carbonate. So this is the two terminals for linear sequence of the amino acid that will be called as primary structure for proteins. So look at this one. This primary structure have N terminal on one side and C terminal on another side. And it's a sequence of the amino acid. You can see here first amino acid, second amino acid, third amino acid, and so on. So what will be the sequence of the amino acid? But there will be one N terminal, NH2 you can see here, and C terminal, C double OH. So it's a linear sequence. What you have to remember, it's a linear sequence as we have seen in this previous slide. You can see the amino acids are arranged in a linear sequence. So talking about the secondary structures of the protein. In the primary structure, you can see here that this is the primary structure. Amino acids are linked together in a straight chain. For secondary structures, these primary structure proteins that are arranged in two forms, either in the alpha helix form or in beta sheet forms. So you can see these arranged itself into the alpha helix. This is the alpha helix structure, this is the alpha helix structure, and this is the beta sheet structure. So itself it may fold itself in either a alpha helix structure or beta sheets type of the structure. And you can see these are the peptide linkages. These are the peptide linkages. These are the peptide linkages. So we will be talking now about the secondary structure of the protein, and we will be talking in details about the alpha helix structure and the beta sheet structures. So first of all, we will be talking about the alpha helix structure. As you can see, this is a spiral type of the structure. It's a spiral type of the structure. You can see having the R groups that are oriented on that side. The R part of the amino acids that are oriented on that side. So you can see here the twist, and the distance between these twists is 0.54 nanometer. It's a very small distance, and each of that twist, that is called as the pitch of the twist, that contain on average of about 3.6 amino acids. So again, the hydrogen bonding is the stabilizing force for these alpha helix type of the structures. So look at this one. The of R part of the amino acids that are oriented outside. So what is that? In the previous lecture we have seen, for example, this is the carboxylic end. This is the primary carbon, and on this side amino group. And on this side H, and on this side this is the R. And we have seen in the previous lecture that this part is almost the same for all the amino acids. Only difference is the R. So the R part of that amino acid that it oriented on outside, and the remaining part, this one, that is actually enclosed here and participate into the bondings. So when we talked about this is the things that are involved in the bondings, hydrogen bonding is there. So that hydrogen bonding that is stabilizing the structure of the alpha helix in the spiral form of the secondary structure of the proteins. So look at this one. This is the hydrogen bonding that is actually the providing the stabilizing force for this alpha helix type of the structures. And as I talked earlier, on average, 3.6 amino acid that resides in one spiral. So from this to this distance, on average, 3.6 amino acid that resides. And what is the distance? An average distance, that is very small distance, you can see 0.54 nanometers. So the average distance from this point to this point in this spiral alpha helix structure, that is about 0.54 nanometers. Another form in which this secondary structure of protein that can be well explained that exists, that is the beta sheets. You can see that how these are arranging itself. The primary structure, this is the primary structure, how that is arranging. So there are two forms of the beta sheets. Either it may exist in the parallel or anti-parallel forms. So both have the different stabilizing action. So look at this one. Each row that is each of one, this one and this one, this is the primary structure. You are now well aware of that. That is representing the primary structure. This is representing the primary structures. So look at this one, how that are stabilizing. This is the CO, this is the NH. So that is making a peptide linkage. This is making a peptide linkage stabilized by the hydrogen bonding. So COO and this NH stabilizing by the hydrogen bonding. And you can see at one end there is a nitrogen terminal and on other end there is the carboxyl terminal. So look at this one that represent the parallel beta sheets and anti-parallel beta sheets. So look at this one, you can see in anti-parallel beta sheets, one sheet that have the nitrogen on this side, nitrogen terminal, carbon terminal, and for another sheet, this is quite opposite to this one, NC. For other sheets, N is here, C is here. So it is a anti-parallel. You can see this is a anti-parallel fashion. But in this case, this is a parallel sheets. The same nitrogen end, carbon end, nitrogen end, carbon end, nitrogen terminal, carbon terminal. So these are the parallel beta sheets. For the tertiary structures of the proteins are actually arranged in a 3D space. You can see here some of the primary structures are available, then secondary structures are available in the form of the alpha helix and beta sheet, parallel beta sheets, anti-parallel beta sheet, then again some of the alpha helix structures. So all of these are arranged in a 3D space. So this is called as the tertiary structure of the proteins. So we can see different types of bondings here in the tertiary structures. You may see the hydrogen bonding, you may see the disulfide bonding, you may see the hydrophobic interactions, you may see the ionic bondings. Look at this one. This is the peptide linkages. You can see here. These are the amino acid, these are the amino acid, these are the amino acid, and these are actually making just like a helix type of the structure. So these are the peptide linkage, and among these peptide link, hydrogen bonding is the forces that is stabilizing these amine linking and stabilizing. So there are some amino acids, sulfur containing amino acids, cysteine, cysteine, just like that. So these are providing the sulfur for the disulfide bonding. Again, these are the stabilizing forces. Sometimes these tertiary structures are stabilized by the hydrophobic interactions. You can see here. So these are the basics for the tertiary structure, and these are arranged in the 3D space. Then quaternary structure of the proteins, these are more complexes, forming the dimers, forming the trimers, forming the tetramers. You can see we have dimers mean there are two structures that are arranged in the 3D space. And you can see how much it's complex, comprising of the primary structure, comprising of the alpha helix type of the structure, comprising of the beta sheet structures. So all of these are the what we have discussed. These are the tertiary structures. So there are so many forms. All of these are arranged. Let's say this is one monomer, this is another monomer. So it's a dimer. And among this, all of this primary structure, alpha helix type of the structure, beta sheets type of the structures, alpha helix type of the structures, then so these are arranged together in a 3D space, forming a monomer. So if there are two monomers, this will be called as dimer. If there are three monomer, this will be called as trimer. If there are four, these will be called as tetramers. So this is the representative figure for the quaternary structure of the protein. You can see the four monomers that are involved. First, second, third, fourth. So these four monomers are involved to make a complex structure of the protein. And you can see here there are some primary structures involved. There are some alpha helix type of the structure that are involved. These are again the alpha helix type of the structure. There are some secondary structure in the form of the beta sheets that are available. So you can see this is representing a monomer. So there are four monomers. So it will be called as a quaternary structures in this case. Thanks for watching the video. If you have some question, you may write in the comment sections. Thank you very much.